If you're a neuroscientist with a penchant to be profiled in the news, one almost surefire way to get there is to study sex differences in the brain, and then make some grand conclusions about how your work proves that men are indeed from Mars and women are from Venus. Some favorites include, male and female brains just aren't in sync, or women need more sleep because they have more complex brains. Hey, I'm not going to argue with that. But if you're a person who wants to know what science can actually tell us about ourselves while avoiding hype and hyperbole, you'd best apply your skepticism and set the bar for evidence way up high when you read a popular press article documenting the latest study on sex differences. There are most definitely biological differences between the brains of men and women. After all, sex hormones dictate some of our neural development, even in the womb, and affect brain function even in adulthood. And of course, natural selection is not the only force determining evolution. Sexual selection has guided us all along the way too. That is, a trait can be favored in a population if members of the opposite sex find it attractive and select their mates accordingly, and then go on to have more babies. The peacock's tail is perhaps the quintessential example. It doesn't make the peacock better able to survive in the wild. In fact, it makes it harder for it to evade predators. But boy, do the ladies like it. And so it has persisted and flourished as a trait. I like to think of musical prowess as a trait that has been the product of sexual selection over the course of millennia. The appeal of the rock star and his legendary promiscuity. And I present, as evidence, Frank Sinatra. His voice might not have helped him take down a lion, but it did win him the girl, many times over. This, of course, is just fun speculation. But it also shows you how easy it is to tell just so stories when it comes to understanding how sexual selection plays a role in evolution. And that temptation is best avoided. So what are the popular myths that neuroscientists studying these differences have to fight? One is the idea that men are simply smarter than women, on average, because they have bigger brains. Well, we've already navigated the complicated relationship between brain size and intelligence, and we've seen that bigger doesn't mean much when you compare individuals. And besides, many women have brains that are larger than those of most men. And we've already seen, in the case of Einstein's brain, that bigger doesn't mean much when you compare individuals, since brains can be more or less densely packed with faster or slower connections. There are both morphological and functional differences in the brains of average male and females. But there are also major differences between individuals of the same sex, and these differences often trump sex. Taking men as a large population and women as a large population, we can discern differences on average. But to apply these differences to individuals is simply ill-informed. There's way more overlap in the distributions of traits in both populations than there is separation between them. And individual differences in brain size, morphology, and function are rampant. So when we talk about average differences between the brains of men and women, we need to remember that any given individual you meet may fall almost anywhere along that continuum. In fact, a large-scale imaging study published in 2015 found that most brains contain both female and male traits. That's true whether the researchers were looking at the numbers of cells in different brain regions, what we call gray matter, or the amount of nerve fibers, or white matter, or the connections between different brain regions. Over the course of the whole data set, they found 29 brain regions that showed differences between males and females. But when they looked at individual scans, only 0 to 8% of participants had all male or all female traits. Most of the brains, more than 90% of them, were in fact a mosaic of both genders. And it's difficult to call a brain male or female just by looking at these features. What's more, when the authors further checked datasets for stereotypically male or female behaviors, they also found that individuals were mixed, with only 0.1% of research subjects showing only stereotypically male or female behaviors like scrapbooking, playing video games, or taking a bath. And yet, we often hear about studies that find significant differences. So how should we interpret these conflicting pieces of information? First, we need to agree on how to measure differences, and only then can we interpret their meaning. In biology, morphological differences between males and females of the same species are known as sexual dimorphisms. The difference between the peacock's and the peahen's tail is an obvious example. But when you scan neuroscientific data for sexual dimorphisms in human brains, you have to consider the variability in your sample. For example, a man and a woman who grew up in the same socioeconomic circumstances, share the same culture, and are of about the same age are going to show more similar performance on a set of cognitive tasks than two men with different educational backgrounds, ages, and cultures. The thing is, the studies that do show morphological differences have to have large sample sizes because there's massive variability in both overall and regional brain structure and function between individuals. Furthermore, age is arguably a greater predictor of brain differences than gender for many variables. Two 22-year-olds of different sexes might have brains that are much more similar to each other than a 22-year-old and a 55-year-old of the same gender. And sexual dimorphisms change with age. Some differences attenuate, others are exacerbated. For example, when you look at cortical thickness, or the amount of gray matter in the brains of men and women, the studies are mixed. Some show more gray matter in women, some show more in men, and many find no differences. Some studies show accelerated gray matter loss with age in men than in women. Others show no such findings. What's more, brain function doesn't always correlate directly with brain morphology. Take gray matter once again. Cortical thinning is a sign of healthy neural pruning during development, and a thick cortex can be pathological, as is the case in patients with schizophrenia. Later in life, however, cortical thinning can be a sign of atrophy and the first stages of dementia. So when we talk about gender differences in the brain, we need to consider the fact that the brain is a moving target. The very strength of this organ lies in its plasticity, which isn't infinite. It is limited by biology, but this variability adds an additional layer of complexity to the issue. Are we talking about gender differences in children's brains, or adult brains, or elderly brains? One nice way of appreciating this complexity is to think of gender differences as belonging to three categories. The first category includes differences that are always present, by definition, and dimorphic between men and women. These are differences related directly to sexual reproduction and sex hormones. They're often well documented and generally not misconstrued by the media, and they are found almost exclusively or certainly predominantly in one sex or the other. Male-specific courtship behavior in birds, like the peacock, is an example. The second category includes differences that fall along a continuum, and males and females have different distributions or averages along that spectrum. Women are more likely to be diagnosed with major depressive disorder, for example, than men, though many men suffer from this disease too. These differences are often misinterpreted, or I should say, results from these studies are commonly overinterpreted with a good just so story. And then there's a third category, differences that change over time. Sometimes men and women are more different on a particular trait at one time or under certain conditions, but more similar as conditions change or time passes. Or the opposite. They start off the same but diverge later in life or under different circumstances. Stress, for example, has different effects on the structure and function of the brains of men and women, but the type and duration of stress, whether it's chronic or acute, can have different consequences on the two genders. If you place rats in an acutely stressful situation, males and females will react differently depending on the type of stress. In one study from 2004, for example, the stress-inducing stimulus was physical restraint for an hour. 2 hours after the restraint, male and female rats were trained on a Y-shaped maze. Male rats were impaired, while female rats showed enhanced learning, even though there was no difference in the amount of cortisol that they had in their bloodstream. In another study, the stressful stimulus was exposure to a predator, a cat, and the task was the water maze. Females performed better overall in a water maze, but both males and females showed impairment after exposure to the cat, with females showing a greater physiological response to the stressor in this case. These studies suggest that male rats fare worse under acute stress, even if they find it equally stressful. But if the task to be learned is to avoid something very aversive, like a shock, rather than approach something appetitive, as is the case when a rat learns to run a maze to get food, males actually fare better than females under acutely stressful conditions. And this difference in learning has a biological basis. Male rats grow bushier or denser dendritic trees in the hippocampus when exposed to intermittent tail shocks. Females show a decrease in dendritic branching under the same circumstances. But these differences take about 24 hours to show up, which might explain why females can outperform males shortly after an acutely stressful event, but the effect of the stress catches up with them later on. So you might conclude that females can weather stress better in the moment, but ultimately suffer later on. But hang on a minute. Chronic stress damages the hippocampus in male rodents and monkeys, but doesn't seem to be as harmful for their female counterparts. The bottom line is that the answer is very complicated. The type of stressor matters. Female rats aren't stressed by overcrowding, for example, whereas males are. As does the task. Is it aversive or appetitive? That's used to test the effects of stress. The timing matters too, chronic or acute in terms of the stressor, immediate or delayed in terms of the learning test. Each of these variables interacts with gender to produce different effects. And we haven't even gotten to the estrus cycle yet. We don't know yet exactly how acute and chronic stress affect neuroanatomy differently in men and women. But we do know that the incidence of disorders that correlate with stress, PTSD and depression, for example, are more prevalent in women than in men, or at least more often diagnosed as such. Now, while mischaracterizing the biological differences in the brains of men and women is not a good thing, there is a corollary of interpreting these data that is even more damaging. When we make the inference that sex differences in the structure or even function of the brain predict behavioral differences, which can then lead to discrimination and prejudice, all founded on a mythical idea that a person's gender can accurately predict brain differences and therefore behavior in a particular individual. But before we get any further in our discussion of how this myth can perpetuate discrimination, we need to agree on some vocabulary. When scientists talk about sex differences, we mean the anatomical and secondary sex characteristics that are dimorphic between men and women and are thought to have evolved via sexual selection. When we talk about gender, we mean the cultural or social construct with which a person identifies. Sex and gender have never really been black and white distinctions, though they have been treated as such by our society for a long time. So I want to be sensitive to the fact that, just like the brain, a person's gender identity and even sex characteristics are not always easily categorized as male and female. And neuroscientists understand this, but the vast majority of the work I'm discussing has been undertaken using participants whose gender identification is in line with their anatomical sexual characteristics. We still have a long way to go before we can understand the neurological and functional differences that might be present in individuals who identify as transgender. There has been some work looking at differences between homosexual and heterosexual individuals in terms of brain structure and function, and detailing these differences is somewhat beyond the scope of this lecture, though I will point out that there is a growing literature suggesting that when we do find brain differences attributable to sex, often the brains of homosexual men are more similar to those of heterosexual women than to heterosexual men in terms of these attributes. The same goes for homosexual women and heterosexual men. But there are several key differences in the brains of heterosexual men and women that are worth highlighting. There are 3 regions in the brain that on average have been shown to be different in men and women, after correcting for body size, that I would like to focus on. The amygdala, an almond-shaped structure that plays a key role in the emotional modulation of memory. The hippocampus, whose job is to lay down new long-term memories and to help us navigate through space. And the corpus callosum, the fiber tract that joins the left and right hemispheres. But you'll soon see that the story isn't quite as simple as we'd like it to be. Let's start with the amygdala, which is generally larger in men than in women. The amygdala helps us remember details of events that were very emotional, where you were when you heard about 9/11, for example. We used to think that the structure, or more accurately, it's a set of nuclei, since the amygdala is actually made up of several smaller structures, was primarily concerned with negative emotions, especially fear. But with more data, there's now good evidence that the region leaps to action when we are experiencing strong emotions, regardless of their valence. And when the amygdala is active, we tend to form stronger and more vivid memories of whatever we're experiencing. You might have heard the term flashbulb memory. This is a memory that seems to be particularly vivid, as though a moment in time has been captured by the flash of a camera. We'll consider the accuracy of flashbulb memories in a later lecture, but for now, suffice it to say that the amygdala plays a role in telling the hippocampus, the region that lays down new long-term memories, to pay attention. There are studies documenting behavioral differences in the recall of emotional events by men and women. Women seem to have a stronger and more detailed memories of emotional events and are able to bring them to mind more quickly. So the idea is that the increase in memory strength that happens with emotion is bigger in women than in men, on average. And while this memory enhancement sounds like a benefit, it might also be one of the reasons why women tend to be diagnosed more frequently with disorders like depression, anxiety, and PTSD. And there's some evidence that memory for things that happen just before an emotional event is worse in women than in men. There's also evidence that the amygdala has different functions in men and women when it comes to sexual behavior, at least. You can easily train a male rat to press a lever that opens a door and gives him access to a receptive female. But if you lesion his amygdala, he doesn't press the bar as much. He'll still get excited if you put the receptive female into his cage, but he doesn't seem to have the motivation to make the connection between the lever and the female. So scientists interpret this work as suggestive of a role for the amygdala in male appetitive sexual behavior. But for female rats, lesions to the amygdala don't seem to have an effect on sexual behavior. And in humans, when looking at sexually stimulating images, men show greater amygdala activation than women, and this activation is accompanied by a greater physiological response. And even when you equate these arousal levels, that is, if you carefully select images that elicit a similar magnitude of sexual arousal in men and women, you still find differences in the functional activation of the brain, including the amygdala. And as we mentioned a minute ago, the average male amygdala is larger than the average female amygdala, even when you account for total brain size. The amygdala is full of sex hormone receptors. So this biological difference can be attributable to the brain changes that happen as a result of sex hormone activity. So the amygdala develops differently in men and women, likely because of the different levels of sex hormones in the brain. The role of the amygdala in sexual behavior is closely tied to the function of another brain region, the hypothalamus, which is the hormone production factory in the brain. And at least one part of the hypothalamus, the part involved in the production of sex hormones, is also larger in heterosexual men than in heterosexual women. And this region is smaller in homosexual men than in heterosexual men. It's one of the anatomical differences in homosexual men that makes their brains more similar to heterosexual women than heterosexual men. We've also already mentioned that there are anatomical differences in a structure adjacent to the amygdala, the hippocampus. As I mentioned earlier, the hippocampus is involved in laying down new long-term memories for things like facts and events. It's also your built-in GPS. In London cab drivers, that 2-year intensive training program in which they have to learn all the streets and landmarks in central London actually results in an increase in the volume of their hippocampi. Have you heard the stereotype that men are better than women at navigating through space? Well, several studies have found that women tend to have a larger hippocampus on average than men. And that might be because the hippocampus tends to decline with age, starting when a person is in his 30s, in men, but not in women, as described in a study from 2001. And we've already discussed how stress can have different effects on the hippocampus in men and women under different conditions. But a large meta-analysis published in the journal Neuroimage in 2015 found no differences in hippocampal volume between men and women. So maybe those other studies that found differences simply didn't include enough participants. You might also have heard that men and women differ in terms of how their brains are wired. Usually when we talk about wiring, we mean how the different parts of the brain are connected. And you've probably heard the idea that the two hemispheres are more tightly connected in women than in men. But on average, women's brains have proportionately more gray matter than men's brains, and men's brains actually have a larger percentage of white matter. And as we've discussed elsewhere, white matter is the stuff that connects neurons. It's called white because the fatty sheath covering a neuron's projections that helps speed the signals from one cell to another looks white. Moreover, when we consider the corpus callosum, which is the largest bundle of white matter in the brain, the connectivity story gets even more complicated. The corpus callosum is the fiber tract that joins the left and the right hemispheres in the brain and is often cited as one of the regions that shows robust sexual dimorphisms. Studies have found that women tend to have larger and more bulbous corpus callosa than men. And this finding has been interpreted by the media and others as showing that women have more communication between hemispheres and think more holistically. This difference was first reported back in 1982. Then, in 1991, a second paper came out indicating that it's more bulbous in women, but more tubular in men, and the total area is the same. Then, a meta-analysis in 1997 found no significant sex differences across 49 studies of the corpus callosum. That one didn't get as much media attention. Finally, a study in 2003 in India showed that the corpus callosum is longer in Indian males than in Indian females, and that it increases with age in males, but not in females. What these studies show is that measuring brain volumes even in the same region is complex, and that variability with age, culture, and other factors muddies the waters significantly. And yet, when those first studies came out in 1982, many scientists and journalists were quick to tell a good just so story. Of course women have greater connectivity between their hemispheres. They think more holistically and intuitively. And of course men's brains are more modular. They look for solutions to problems and focus on one thing at a time. But the truth is much more complicated and nuanced, of course. Some of these differences in connectivity have also been used to promote the left-right brain myth. That because women are supposed to have greater interaction between the hemispheres, they have greater access to the non-dominant, intuitive, and emotional right hemisphere, while men are more dominated by their logical, analytical left hemisphere. We address some of these holes in this type of reasoning in our lecture on brain hemispheres, so I won't revisit them now. But I would like to take a moment to address the question of whether men and women process emotions differently. Women seem to be both more expressive of emotions and better able to read emotions in others. But men and women don't seem to be that different in terms of their experience of emotions. In other words, they feel joy, sorrow, pain, and so on in much the same way. So it's not true that women are more emotional than men in general. Instead, women are more emotionally expressive, and the types of emotions that men and women experience more frequently might be different. Men seem to experience anger and aggression more often than women, who seem to cry and smile more frequently. And the social context seems to matter a lot when it comes to seeing these differences. The presence of another male makes it less likely that a man will express emotions, and women tend to show anger more directly in private, but more passively in public. So these differences in emotional expression seem to be driven largely by culture. Men are evaluated more negatively than women when they are seen to be expressing sadness, fear, or shame. And the opposite is true when women display anger and aggression, particularly in childhood. Sex hormones might play a role, too, in regulating these emotions differently in men and women, though we can't discount the strong effect of culture. But besides helping us understand or misunderstand each other, depending on how you look at it, there is a need for more research on sex differences in the brain. And that's because these differences, when they're real, have direct implications for our health. You might think that that difference is limited to disorders closely tied to reproductive organs, but that's not true. There is a difference in prevalence, for example, for many psychiatric and neurological disorders between the sexes. And in order to properly treat men and women with these disorders, we need to understand the differences in their neurobiology. One prime example is Alzheimer's disease. Women are more at risk than men at contracting the disease, and a recent study showed that the effect of AD pathology is different in men and women in terms of their cognitive function. Men can show much more evidence of Alzheimer's pathology, the plaques and tangles, without showing symptoms of the disease or being diagnosed with it. Whereas for women, it seems that it takes much less pathology for symptoms to appear and for a diagnosis to be made. Similarly, the genetic risk is also different. A single ApoE4 allele, the genetic mutation that is related to Alzheimer's disease, can have a much greater effect on the atrophy of the hippocampus and the resulting memory dysfunction in women than in men. We can find sex differences in people with schizophrenia as well. Men with schizophrenia show larger ventricles. These are the spaces in the brain that contain cerebrospinal fluid than healthy men. But women with schizophrenia don't seem to show this anomaly when compared with their healthy counterparts. And herein lies the need for more research into sex differences. Men and women might react to the same conditions in different ways. They may then show biological changes that are not the same, and we need to understand these differences if we're to properly diagnose and treat diseases. Sex differences can show up in disease prevalence, in prognosis, and/or in response to treatment. And yet, there's a very prevalent bias in medicine to focus on male patients and male animals. Why? Well, amongst animal researchers, the argument has been that unless there's a clear evidence of sex differences, male animals are easier to deal with than female animals because they don't have to worry about the estrus cycle. And the hormonal changes that accompany estrus do affect animal behavior and cognition. So females are underrepresented in much of the medical research. This is problematic because side effects and effective dosages of many drugs can be very different between men and women. One example is Ambien, the drug used to help people sleep. In 2013, the FDA told the manufacturers of the drug to cut the recommended dosage for women in half. But the drug had already been on the market for 20 years, and this entire time, women were told to take far too much. How did this happen? Much of the early research, including work that was designed to find effective dosages, was conducted exclusively on men. What's more, the effects of age on brain volumes are also different in men and women. Men seem to lose more brain volume overall with age than women, but women lose more volume in specific regions like the parietal lobes. So there's a strong argument for correcting this gender imbalance in medical research in particular, so that women can benefit from treatments that are designed for their bodies too, and not just for men. But the dark side of studying gender differences in the brain is that they are so easily misinterpreted, with quaint and potentially damaging just-so stories. We've barely touched upon the fact that even assigning gender is more complicated than it seems. So some of these findings might not hold for individuals whose gender identity doesn't match their anatomy or whose sexual preferences are not exclusively heterosexual. Human behavior is complex, especially when we're discussing higher cognitive functions like decision-making, remembering, identity. There are many different paths to achieving a full understanding of our own brains. But remember that differences in brain morphology are not always reflected in differences in performance. They might just lead to different strategies towards finding a solution. And this diversity is beneficial in almost any environment.